Swayam Prabha. Digital India. Educated India.
Right. So this is the complexity of the different amino acids residues that I shown before. So this is the structure that I had shown you, right? And if you actually zoom in and look into this structure, you can see that these are nicely packed, right? You have one amino acid side chain. This is the side chain of, say, let's say this is a lysine residue, or this is isoleucine or a valine, and this is actually nicely interdigitating with respect to the other residue. So these are actually packed like what you would expect in a puzzle. You can't have any gaps in between. So you'll need to be optimally packed. And if you need to be optimally packed, what it needs to be also happening is that if you introduce a mutation here, and you know what a mutation is, change in the sequence of the protein, some specific substitution happens, what can happen is that this particular, say, side chain cannot interdigitate properly with this particular case. So the protein will not be able to adopt this particular structure. It will unfold, and hence it will not carry out the function that is required to carry out. So this is generally what happens when you have mutations. And you know that mutations happen all the time within the cells. Right now mutations are constantly happening. Some mutations are beneficial, and many mutations are actually deleterious, because they don't give rise to anything very interesting, right? So in reality, well, I'll take a step back here, and I showed you that this is the structure of the protein, but this is what you might have seen in your biology textbooks. I don't know what it is that you saw, and, or even if you take some advanced biochemistry books, this is what you would see. But in reality, this is not the way it is there within the cell. Within the cell, what happens is that you ask this particular question: if they don't like to interdigitate very well, if the interactions are not very strong, then this will be very weak. And if this is very weak, then it can actually go back and forth like this. So this is something that happens all the time. So if you really look at a protein within the cell, this is the way it will kind of be wiggling around. All right. This is actually the folded structure that you saw. It will be doing all kinds of stuff. Okay. This is called the dynamics of the protein. So once the protein folds, it doesn't answer the question completely. It depends on what kind of motions these different regions do, that is more important. And why do these guys move around so much? Because they are not very happy with the interactions that they are making. There are some regions of the protein that fold or pack very well. For example, you can see that this particular helix is nicely folded. This is this spring thingy which I said. It's nicely folded all the time. But you can see that this guy keeps folding and unfolding, and you would see that it folds, unfolds. Yeah, it unfolds, it does something weird, right? It goes back and forth, and that's because it's not very happy in the environment it is in. And this is also very important. So this is called the dynamics of a protein molecule. It is not as simple as what you see in the structure. So don't trust your biology textbooks. Okay. So there's more to it. And you do biochemistry and do higher studies and whatever, there are many things that you would learn. This is actually fun. You can actually model these guys. You can actually take the protein sequence and you can put this in a computer. You can actually add water molecules around it, and you can actually simulate the system. And this is basically something called the molecular dynamic simulations. But of course here, what I know is that this is the final structure of this particular sequence. When I have this sequence, this is the final structure it adopts. Once I put the structure and I can simulate using simple Newton's law of motion, I can actually simulate all these different motions, as which I can just plot it again. I can just run this again, and this is what happens. Okay, this is of a folded protein. So once a protein folds, these are the different motions that take place in a protein molecule. And so you should not be kind of taking the structure that you see, which is very static, as what is a representative of what happens within the cell. Okay, there are many motions that happen in a protein molecule, which is not the reason that I want to get into. There is more to it, in the sense that the overall folding of a particular protein has a lot more to do with something called the entropy of the chain. If I want to go back and answer this particular question, and I told you here that it is easy to move around. So for example, what I mean by that, I can fix this particular plane. I can actually move this around and move it. for every angle, for a fixed value of phi, I can have multiple angles of phi. Similarly, for a fixed value of psi, I can have multiple angles of phi. So if this is the case, and if you have a 100 residue protein, how many degrees of freedom do you have? How many would that be? So let's say I have a 100 residue protein, okay, and every residue, let's say, has 2 degrees of freedom. So then 198, no. Every residue will adopt a certain configuration, so it will have 2 degrees of freedom. Then, so think of it as some mathematical problem. So that's easier for you. 100 power 2. Don't worry about the end effects. Let's not worry about it. Keep it simple. 2 degrees of freedom, it will be 2 power 100. Right? Every residue will have 2 degrees of freedom. And so for every other residue, you will need to consider all the different configurations that are possible, fixing one. So you will effectively have 2 power 100 different states of a protein. And what I mean by the states of a protein is that you can have 2 power different conformations of the protein. The structure, the XYZ coordinates, or even each and every atom can be very different. So which means I am making a simplistic assumption that there are only 2 degrees of freedom. But you can see here that I can fix the phi and I can have multiple angles of psi. So I am discretizing it into only 2 degrees of freedom, but technically you can have a continuous variable that goes all the way 60 degrees, 60.1 degrees, 60.7 degrees, 70 degrees, 70.1, 70.2 and so on and so forth all the way till 360 degrees. So I can do this. So which means there are many degrees of freedom associated with the protein chain. And what does it mean? If a protein is synthesized in the ribosome and it's completely disordered, meaning it does not have a very specific structure, it is like a beads on a chain that I had shown you before. If it actually looks like this, right? How does it know that this is the final structure that I need to adopt? The structure that I had shown you on the left or right, whatever it is, how does the protein chain know? And how quickly can it sample it? Let's say, okay, it's length of the protein, let's say 100 residue protein. How quickly will a protein able to sample? Typically, a protein will fold, a 100 residue protein will fold within a minute. Meaning from the time of synthesis in the ribosome, it will actually fold to the final 3-dimensional structure in a minute. But how does the chain know that that is the final structure? That's the puzzle. That's the question. So how does the chain know? It doesn't. So for example, what is it? It's more stable in that particular state. So if it needs to find a state which is more stable, then it needs to sample each of the different states. Yes, for example, again, I ask you guys to go from the IIT Madras main gate to the Gandhinagar beach, right? So now I ask you this particular question. If you are new to this particular city, would you be able to answer the question, which one is the fastest route? So then the protein chain, does it have these visions and thoughts and say, hey, I see an electrostatic nice positively charged guy out there, let me go and interact with him. I'll make a nice electrostatic interaction between them and then I'll fold. It will, or potentially, or it could try all possibilities. What you can do is that now I give you this particular question. Now you start today evening 5 p.m. or 6:30 p.m. I don't know, whenever this presentation gets over. So you go there, you start sampling all the different routes one by one, and then finally you come up with some number of routes. So now and then you come back to me and I say, hey, which one is the fastest route? So now what do? So you can't take distance into consideration. Why is that? You need to also worry about the traffic. Some routes will have some signals, shorter the distance, but you might have more traffic signals out there, which means it will take longer to reach. So you need to worry about all of these things. But a protein chain, when it's like this, in this particular conformation, how does it know that there is some structure that I can attain? How does it know a priori? That's the question. it is encoded in the DNA. Yes, the sequence of the protein is encoded in the DNA, and you get this particular protein chain. But how does the protein chain know that it needs to go there and not anywhere else? But how does it know? No, you can take a purified protein, you can put it in a test tube, and it will still adopt the same structure. And the only way the protein, if you can think about it, there's something called the Levinthal's paradox, which I'll take you to in the next second. Right, is the fact that an unfolded chain can actually adopt many different conformations in an astronomical. I can't even count, or no one can count for that matter. You can count, but you will spend your lifetime and a million times over as well. Right? If you are going to have 3 possible conformations, or let's say you fix the phi and you adopt 3 different psi values, and you can have 3 power 100 possible conformations. Even if a protein can sample 10 to the power 12 conformations per second I'm talking about, it samples 10 to the power 12 conformations or different orientations of the side chains or main chain with respect to time, it will take about 5 into 10 to the power 35 seconds, which is 10 to the power 28 years, which is effectively greater than the life of the universe. So the protein cannot arbitrarily sample all the possible conformations that it can take and then identify one conformation which is the most stable. And what does it mean to be most stable? At that particular conformation, it has all the nice favorable interactions on average. Meaning all the positively charged residues are connecting to a negatively charged residue. All the hydrophobic interactions and the interdigitation of the different residues are perfect. So the protein adopts this particular configuration. But to reach that particular conformation, it needs to sample all the different states. So how does it go about doing it? Right. I'll then go back. So for example, this is the constraint that I told you, is the fact that any protein in a E. coli has a doubling time of 20 minutes, right? Within 20 minutes, you'll have the daughter cells that are being formed, and these will again divide in another 20 minutes, you'll have the other daughter cells that are granddaughters and so on and so forth, right? So in this particular case, a protein will need to actually fold within a 20-minute time span for it to be useful for the cell. If it is going to take longer, it is never going to be useful for the cell. So, but in reality, many proteins fold within a minute. But if you do the calculation as per the Levinthal's paradox, what you see that it will never be able to fold. So what is missing here? That's the puzzle. So what the heck is missing here? That's the question. That's the way. Maybe invigorate you a bit more. Yeah, why? How is it able to fold? Any idea? Okay, that's a good point. There is no covalent interaction. So it will be primarily a covalent interaction has already happened through the peptide bonds because the ribosome kind of stitches together the amino acids. Yes, the electrostatic interaction can form. Yes.
So they happen first. So in confirmations you can do that.
Yes. Do you understand what she's saying? Yeah. Well, anyway, so let's hear from the others.
Maybe it doesn't have to go to the most stable folding pattern that is possible, but rather a pretty stable one, and that is why we see the even after the folding we see how it keeps changing and like keeps vibrating. So it's not at the most stable folding, but it is stable enough for it to be used.
Yes, but how does it know that that is the final structure? That's the question. So initially you don't have any structure. It doesn't have this helix, doesn't have a strand, doesn't have this spring or the arrow that I shown you before. It doesn't have those structures. It is basically like a linear chain, let's say. But now, if it needs to adopt a structure similar to what I had shown you before, then it needs to sample all structures and rule out one by one with a brain of its own, if it has one, and then find out that structure that results in most stable or the highest stability. But that is not possible because it will take about a time which is more than the life of the universe for it to really happen. So, but what can happen? You have a fair point. I'll come to it in a minute.
Rather than looking for the most stable structure for the entire protein, maybe it's like a couple of adjacent amino acids.
Yes.
Just go to the most stable adjacent configuration.
Yes.
And then if every amino acid on the chain does that, eventually it will get to the...
Yes, that's exactly what happens, in the sense that as you also said, basically what's going to happen is that the amino acids which are next to each other will try to interact 2 or 3 amino acids. So if I ask you to pass on the information from you to the person out there, you're not just going to keep shouting. So what you will do is, in a classroom, you can shout technically, but I'm saying here, you will pass it on to the next person, this person will pass on to the nearest neighbor, and the nearest neighbor will pass on to the next nearest neighbor, and so on and so forth. This effectively happens in proteins as well. So if you have some charge residues which are nearby, or 2 residues which are happy to interact with each other nearby, then some local structure can form. If I can draw it on the board, basically what you can have is that, let's say this is what happens this is the way it will look once the protein is synthesized from the ribosome. This will not really be good. So basically what it does is that it will form some structure here and everything will be completely random. Now these will have some favorable interaction, some hydrogen bond, some electrostatic interactions. So now you can see that the protein need not sample all the 3 to the power 100 conformations, because some region has already become structured. Only these guys will need to sample. Right now some region here can adopt a specific conformation, let's say. So this can go to a state that looks like this. And effectively what you can have is that these 2 guys coming to meet each other, and they can form a structure that looks like this. Then effectively, then the entire protein can fold, form a 3-dimensional structure. So basically you can see the regions that are close to each other can interact first, and once these guys interact, that will stabilize that particular structure, as he said, and then effectively they keep iteratively searching through the conformational space iteratively. It's not going to be in a very directed way. I'll come to it in a minute as well. So you will have a final folded structure, right? So this is the potential way the protein molecule can fold. But what information is going to tell that the local interaction will form first and a non-local interaction will form later? By non-local, I mean regions of the protein that are very far in sequence. Local means regions of the protein that are close to each other in sequence. What information does it have? So effectively, this is the grammar that I have been talking about. So if you pattern the amino acids in such a way that some local interactions can form quickly, what can happen is that you can have multiple regions that can actually fold independent of the other regions. And then these guys can come together and they collapse and they form a nice folded structure. But if you do not pattern the amino sequence in a way which is reasonable, then if it is a random polymer sequence or a protein sequence, the patterning will not be right. So the protein will not be able to form favorable interactions very quickly. It will take forever to fold. So those regions will not be able to fold. So now all these mutations and everything that happen in the DNA, what they do is that they ensure that the patterning of amino acid is just right, so that the protein is able to make all these interactions in a reasonable time frame and adopt a well folded 3-dimensional structure. Okay, so this is really what happens. So what I mean by patterning is that you need to have specific amino acids in specific positions in the sequence. If you don't have it, then it will take much longer for the protein molecule to fold. Okay, this is exactly what happens when it comes to proteins. In the sense that you will need to know, right? So you will need to effectively design the sequence through natural selection, meaning multiple rounds of mutations and so on and so forth. And the sequence information, of course, is present in the DNA. So the DNA tells, okay, these are the amino acids that need to be right next to each other. So the information is already present, and so the protein folds. But now what we are trying to do is that we are trying to understand what is the grammar. So we are doing the inverse of what you guys have been taught. The sense that you are taught A, B, C, D, E, and then you taught to put together the letters and make sense out of these letters and make them words and words to sentences and so on and so forth. But right now, if I go into any protein sequence database, I will have only the final constructed collection of letters which corresponds to the protein sequence. I do not know how they make sense. and what is the sense that is already buried in them. So that is something I do not know or nobody knows. So, but there are few people who know. I'll come to it in a minute in some ways, right? So, are you able to understand this part? So, basically you need to arrange the amino acids in such a way that they actually fold in a reasonable amount of time. So, local interactions will form first, non-local interactions will form later. This enables the protein to fold very, very fast. But if local interactions need to form first, then you need to have certain amino acids next to each other in a favorable interaction energy, and that will result in local pockets of stability in the structure.
The local sequences are decided by the DNA.
Yes,
And
the entire sequence is decided by the DNA, but again it's all the pattern. So it will also come down to the length of the protein. Longer is the length of a protein, longer is the time it will take to actually fold. Shorter the protein or smaller the protein, which is 40 amino acids or 100 amino acids, it will fold faster because it can sample things much faster. Right. So how do you go about doing this, right? So this is something that I said, this is the 20 amino acid alphabet. So, and if you really think about it, there is another combinatorial complexity associated with it. In reality, not every patterning will result in a folded protein. Only a certain patterning will result in a folded protein, because in reality you can have, for a 100 residue protein, you can have 20 to the power 100 different sequences. Only some of them will fold, because only those guys will form the stable interactions in a reasonable time frame for them to adopt the structure that I have shown before. Otherwise they will not. Okay, so that is something that you need to remember. So, you can have many sequences, and every sequence will have 10 to the power, 30 different conformations that are possible. So, you can have 10 to 20 to the power 100 sequences, and you will have 3 to the power 100 conformation per sequence. So, you can imagine the complexity associated with the problem. In reality, if I ask you to say, hey, go ahead and design a protein that adopts this particular shape, you will need to basically navigate all these 20 to the power 100 sequences. For each sequence, you will need to navigate 3 to the power 100 conformations to get to the final structure, which is technically not possible. Nobody can do that, at least at this moment. Okay. So you can imagine this. So if you can pattern them in some ways, it will make sense. If you pattern them slightly differently, they will not make sense. Can you understand? So if I have these letters, they will result in proteins. Yes, but they can also result in something else. What else? This is an anagram, obviously. Proteins, yes. If you put them together, it will be proteins. It makes sense. Then, okay, now it's actually English word that you have. But what other English word can you make? Well, okay, so it's taking a bit longer. You can also make more pointers. You can, so basically you have the same sequence, same composition in some ways. You have the same letters, but the way you organize them can result in a slightly different word, and some words will make sense, some words will not make sense, or rather, majority of the words will not make sense, and a few words will make sense. That in reality what happens? So basically the evolution of DNA and hence this protein sequence has happened in a way that some amino acid sequences are actually making sense, they fold in a reasonable time frame and are able to function. So this is exactly what happens. So basically the patterning of amino acids here, in this case the letters as a function of these words, right, is what really matters. So this exact same feature is actually exploited by nature. So the nature goes about shuffling the amino acids one after another iteratively, and it has already done this. That's why we are here and discussing about this. So it has done this already, and we have the final answer. Now you tell this baby, a 1-year-old or a 2-year-old to read this. They might be able to, a 3-year-old may be able to read all these letters, but for them to make sense out of this is impossible. Right? And we are at that stage actually. Despite whatever advancement we have all the protein sequences, all this multiple, the genome sequencing that has happened. I'll be finishing next 10 minutes. There's another talk. What is it? What time is it? 5:30 is it? I'll try to finish in the next 5 minutes, max. We started a bit late. Yeah. Right, so basically what does natural selection do? It basically chooses a set of letters, such that they can be spelled. And in the context of protein folding, what it means is that it chooses a set of letters such that it can fold, adopt that specific structure, and also fold in a reasonable time frame. It might, some structures will be folding in 300 hours or 1000 hours, but that will not be productive at all for the organism. You will need to have something that not only folds but also folds fast. So that's what the nature does. It patterns the amino acids in a way that it is able to fold. If you have a small collection of letters, you will have a smaller protein. If you have a larger collection of letters, you will have a larger protein. And if you are able to understand the grammar, you can write paragraphs and you can write these entire multi-assembly complexes that I shown before. Right? You can have these big structures simply because you know the patterning of amino acids. And I'll just skip this part. In reality, this is what happens. If you have a protein chain that looks like this on the left, you can actually come up with something called the landscape, just like what you have in the mountains that I just showed you before I skipped. What you see is that a very complex landscape that you see. Similarly, you can construct something called the energy landscape here, and for every conformation, you can plot it on particular space here, point here. So you will see that as the protein folds, it will start moving around, and it will be randomly sampling states. You can see it's wiggling, jiggling, and now you can see it adopts some favorable state here, and you can see this helix is being formed, and then it folds down and it adopts a nicely folded structure. So this is the way it actually folds in principle. So if we can run this again, it starts in a completely random arbitrary configuration. It keeps sampling many different states, adopts some local conformation as you can see, and there is some local energy minima here, and then it reaches some kind of a global energy minima that happens. So the patterning of sequence is in a way that there are multiple minimas, so it doesn't get trapped in some of these things and effectively escape and come down to the global minima. So the nature has done this already. Natural selection has done this or achieved this already. What we are trying to do is to understand what is going on, and we are trying to understand something called the protein folding code. Meaning, if I give you a sequence, can you predict what is going to be the final structure? That is something not completely solved. There are some nice interesting puzzles that you can work with. There's something called Fold It, which is a computer game that effectively exploits the intuitive understanding that we have, and you can actually play and you can move amino acids around and you can actually see whether it adopts a folded configuration or not. So you can just write this down and you can have a look at this. Can I move to the next slide? 2 more minutes, I'll finish. Right? Can I move on? Right. So one is that there are two ways to pose this problem. One is that given a sequence, can we predict the structure? I give you a 100 amino acid sequence. Can you tell me what is the final structure? And something called the mechanistic details. I'll come to it in a minute. And given a sequence, can we predict the structure? This has already been solved by some program which is called the AlphaFold, which is a deep learning. Have you heard of AlphaGo? I think, yes. It's basically, this game called, what is it? Yes, so using some machine learning algorithms, you can actually predict this. So this AlphaFold 2, which is basically from DeepMind, was able to solve this problem of protein folding in terms of given a sequence, can you predict the structure? So now people don't need to do many experiments actually. So what people can do is feed in the sequence into this particular server, and you can actually get the structure in the next 2 minutes. That's all. So the problem, what is the way it's solved is by using something called the machine learning algorithms or deep learning, and you actually predict. So you learn from the patterns of the sequences that are already present and how are these patterns on the sequences connected to the structures that you see. So you learn these patterns using millions of parameters and you kind of predict the structure, and this has already been solved. This happened just about 2 years ago. Alright. So but the more interesting problem is that now I give you a structure that looks like this. One strand, a second strand and a third strand. Now I ask you this question, how many different ways can this particular protein fold if I give you 3 secondary structure elements, meaning there is 3, 1, 2 and 3. How many different ways are there? Meaning you can have the first strand forming, then the second strand, then the third strand, or it can be first, third and then second, or it can be 3, 2, 1 or 3, 1, 2, whatever it is, right? So how many ways? 6. So what is how do you write the combinatorics here? What factorial? 3 factorial ways in which you can form. If I give you a protein which has much bigger structure, you can have 10 factorial ways. It is same as what you would say when you are going to go from, say, the IIT gate to the Gandhinagar beach. If I give you this and I say, okay, figure out what is the most efficient way. So how would you do it? Basically, this is what happens in a protein chain. You have the sequence is already told, is kind of designed by nature. The protein will be able to fold a three-dimensional structure. Let's say this is where the unfolded state is, this is the folded state, and you can have many different routes. You can have many different ways in which it can fold. It need not go through a single specific route. Which route it takes will depend on the prevailing conditions, that is the cells facing, either due to stress or due to the presence or absence of certain ions and so on and so forth. So effectively you can build many combinatorial problems through this. You can have, say for example, strand 1 forming first, then strand 1 and 2, then 1, 2, 3, or 2 forming first, then it can form strand 1 and then strand 3 and 1, 2, 3 and so on and so forth. You can have all kinds of combinations that are possible. So it is not just the folding problem, meaning given a sequence to predict the structure, there is more or tremendous complexity associated with the number of ways a protein can fold as well. So every time there is a certain level of combinatorics that you need to invoke in terms of protein sequence space, in terms of the number of configurations that a protein chain can sample, in the number of ways in which it can sample to reach the final folded state. Right? So there are many different unanswered questions. I would not kind of bore you with this, but I will stop here because of the time constraints that we have, but I can go on for another 2 hours. So, but I'll stop here. But any questions on this? Yeah.
Is the folding of proteins a purely like deterministic process? Can we guarantee that if we start off with the sequence, it will always end up with the right structure?
If you have the right sequence, you can say that it will go to the fully folded structure. But if you have the sequence which is not designed either by you or some computer or by nature, then you wouldn't be able to guarantee. So the only way you can guarantee is that if you are able to sample all the different states and say that there is one state which is the most stable state. But you can have multiple states or multiple different three-dimensional arrangements, they can have the same energy. Then you can have, for the same sequence, you can have multiple different conformations. That's possible. But so you need to know the right sequence. So the sequence is what we don't know. How it has come out to be that is encoded in the DNA.
If there's third base degeneracy in a codon, the same amino acid is formed, but does that third base degeneracy affect the folding of the protein?
It has been looked upon earlier, and there is a degeneracy associated with it. You can have slightly different amino acids that can be coded, but this will not affect the folding so much. It has not been shown so much. Any other questions? You can actually use simple models to actually look at the ways in which a particular protein molecule can fold. Just like what you have here, you can effectively imagine having a landscape where you drop a ball at the top and you can see the number of routes it takes to actually fold to the bottom of this particular funnel, and you will see that there are many different rates. If you're skiing through a complex mountain or a landscape, you will see there are many ways to reach the bottom. A same protein chain in an abstract landscape will actually take these many routes to reach the bottom of this funnel, and this bottom of this funnel will effectively the fully folded state, which has the lowest energy or the most stable state. I'll stop here, but anyway, there's some questions. No, the alpha fold, what it does is that it will take in a sequence and it will predict you nearly accurately the final three-dimensional structure. But what it has learned, it's machine learning, which means, and deep learning, which means that it tries to fit in multiple parameters to reproduce the existing database that it has already been working on, and it tries to understand the correlations in some ways. We don't know what it understands, and then it predicts the three-dimensional structure. So in reality, if you want to understand the protein folding code, you need to have a physics-based algorithm. or a chemistry based physico-chemical principles that you need to understand, which means that you need to know why certain amino acids are right next to each other, because they favor some interaction and because of which they are able to fold in a reasonable time frame. You need to understand the logic there. That logic does not come out through AlphaFold. AlphaFold only predicts the structure given a sequence. It doesn't tell you anything else. You can change the sequence, it will predict a different structure. But why does it predict a different structure? AlphaFold will not be able to answer that particular question.
Sir, one more question. We saw that the protein can form that last final stable structure through many routes, right? So why does it take the complicated route while it can actually take the simple route?
What is the simple route?
You showed various structures in the this one, sir.
Yeah.
Like the complex one, why does it have to undergo all those transitions before while it can actually go with the first diagram?
So let's say I tie your eyes and with a black cloth and I ask you to go to the IIT gate now. What are the chances that you will go there in a reasonable time frame? It's going to be extremely challenging. You will get there eventually, but it's going to take a lot of time. So similarly, a protein chain, if it is going to start from particular state to reach to final configuration, it will take a lot of time in some ways. I mean, in the time scale that you can consider in microseconds to milliseconds, it will still fold. But there can be many different routes. This results in a faster folding. Some routes will result in slower folding, and that's what happens really. So there will always be a randomness associated with the process. It will always be slightly different route every single time.
Thank you, sir.
Yes. Mutations in the genome will affect the amino acid sequence. When the amino acid sequence changes, the patterning is something that is being changed. When the pattern changes, if it is not a dramatic change in the sequence, meaning you take a hydrophobic residue and put a charged residue out there, then everything is messed up. Right? But if you actually take a larger hydrophobic residue and put a smaller hydrophobic residue, then things are maybe still fine and the protein will still adopt the three-dimensional structure. But it might not be that stable, because whatever sequence that you have right now correspond to the most stable ones. There are many exceptions, there are many different things that I would not be able to do justice to in just about an hour, but I'll just use that as a starting point. And if you have questions, you can always contact me. Yes. I mean, the number of ribosomes are on the lower side, then you will have the synthesis of proteins will take longer. Excess of DNA should not be. It depends on how many mRNA that you have, really. The DNA does not matter. How many plasmids that you have, that doesn't matter, really. Any other questions? I mean, I will stop here, and I don't know how to share my email ID. My name is Adi. You can find me, my name at the biotech website, biotech department website. So if you're interested, if you have questions, you can always mail me, or you can tell me what it is your question is, and I can try to answer you over a phone call, or you can come and meet me in my office. Okay. All right. I'll stop here. Thank you.